So I know that you're familiar with most of the concepts of cellular aging, but I thought that it was a good idea to put together a lecture that summarizes all of the different components of cellular aging. So let's begin by looking at how we define aging. Is aging growing up? Is aging growing old? And then what is really old anyway? And how do we actually define aging? And as it turns out, there isn't really a solid definition of how we define aging, but I'm pretty sure we all have a good concept of what aging looks like. So let's think about the idea that it's an intrinsic process where we have interactions with environment, which could be things like sunlight and toxins and the things that we consume in our diets that cause changes in structure and function of our cells and all of the molecules within them. So if you could think for a moment about all of the things that you know that could contribute to cellular aging, what sort of things would you come up with? Well, we can categorize all of these things, these physiological traits of aging, into 9 different categories. We start out with genomic instability, and I'm sure that already you could put a number of things in that category. My intention in this lecture is to put all of those things in those categories or in these categories for you so that you have it all together and are well prepared on the topic of aging for your exams. So in addition to genomic instability, we have mitochondrial dysfunction and telomere attrition. All of these should be pretty familiar topics, right? We have epigenetic alterations, loss of proteostasis, altered intercellular communications, cellular senescence, as well as deregulated nutrient sensing and stem cell exhaustion. So these are 9 categories of things that can or that do change as cells age and lend themselves to some of the phenotypic manifestations of aging. So we will move through these categories fairly rapidly as I summarize some of the things you already know quite a lot of details about. When we consider genomic instability, we are going to consider that we accumulate genetic damage throughout our lifetimes. So in the form of DNA lesions, through mutations, through environmental interactions and such. So we have both nuclear DNA and we have mitochondrial DNA to consider in this realm of accumulating genomic damage and thus instability of genes and perhaps interruptions or changes in how genes are transcribed and translated. Also, we need to consider the nuclear architecture, so all of the components that go into the nucleus and keeping that system of DNA transcription working efficiently. So when we consider genomic instability, we have both exogenous threats, things that impact it from the outside, as well as endogenous threats. Outside influences can be physical influences or chemical influences in the form of things we are exposed to during life, as well as biological influences from within ourselves and outside of ourselves. So in our endogenous threats, we really are now considering replication errors, spontaneous hydrolytic reactions, as well as damage that can be done by reactive oxygen species. So all of our metabolic processes, cell respiration, the final electron acceptor is oxygen, right? And you are familiar with the idea that oxygen, when we split the O2 into 1/2 of an O2, it is highly electronegative and can run around and do all sorts of damage to our DNA. And as it happens, it's kind of paradoxical. We require oxygen to live, but oxygen in the end is potentially what actually leads to aging and eventually death. So something to roll around a little bit there. So let's look at Parkinsonism versus Parkinson's disease. Parkinsonism is a syndrome of Parkinson-like symptoms, right? That's something that you need to keep straight. Parkinson's disease is not the only cause of these types of symptoms. So we consider Parkinsonism separate from Parkinson's disease. There is primary Parkinson's, or primary Parkinsonism, which is a result of Parkinson's disease and the classic Lewy body and neural degeneration. And then other versions of Parkinsonism we consider as secondary Parkinsonism, so things that develop from non-Parkinson's disease syndromes. So when we treat Parkinson's disease, there are a number of different options. First of all, L-DOPA. Historically, L-DOPA has been around for much longer than it was actually used for treatment of Parkinson's. It was initially used in the treatment of heroin withdrawal symptoms, and it turns out heroin withdrawal symptoms have some of the same characteristics and difficulty of initiating movements and tremors that we see in Parkinson's disease. And so someone thought, let's try this out on Parkinson's patients, and it worked. So that was great progress. However, it didn't work forever, right? The effect of treatment with L-DOPA, even with increasing doses, didn't resolve the symptoms. But initially, L-DOPA on its own would relieve some of the early symptoms of Parkinson's disease. Now, how does L-DOPA work? It turns out L-DOPA, or levodopa, is a precursor to dopamine. So it also crosses the blood-brain barrier, so it can be taken orally, crosses the blood-brain barrier, at which point it is converted into dopamine. Now, it can also be converted into epinephrine and norepinephrine, which can have its own issues. But the dopamine here is essentially adding extra dopamine so that the lower number of neurons have more signal being thrown at them and can make some signals, and this works. Dopamine functions return, and things look good for a while. But the issue comes in that L-DOPA also can be converted in the peripheral nervous system, and here perhaps the epinephrine and norepinephrine create some adverse effects. So we have additional dopamine in areas and norepinephrine and epinephrine in areas where it's not necessarily needed. So in order to alleviate some of these symptoms, what we now do is co-administer this with, or L-DOPA with dopa decarboxylase. And dopa decarboxylase will prevent L-DOPA from being turned into dopamine, and thus norepinephrine, epinephrine, and subdue some of the effects that we see in the peripheral nervous system. So these days, it's very common that L-DOPA and something like carbidopa are prescribed together for patients in experiencing Parkinson's disease. So a couple of treatments that you should be aware of. Now, another treatment that has got some pretty cool history behind it is with deep brain stimulation. Initially, well, this is basically still what happens. We'd open up the brain. These super surgeries are actually fascinating to watch. They will enter the brain with an electrode and drop it into the substantia nigra and stimulate the neurons in the substantia nigra so that they secrete dopamine again. So we're reactivating, re-energizing neurons of the substantia nigra. And it when this was first done, it was pretty amazing surgeries, because you would have someone with an open brain or and electrodes going into the brain and people controlling movement, and it was kind of wild. Anyway, now it's a little bit more common to see these deep brain stimulation technologies, so much so that we now attach them to a pulse generator that can be implanted in the chest, and much like a pacemaker works on the heart, this pacemaker works in the brain with a constant pulse, causing the neurons of the substantia nigra to secrete dopamine. And so this, in a lot of cases, has been quite helpful. Although it's not completely effective, it does have some good possibilities and potentials. Now, after this sort of research was done, they came around and started looking at stem cell therapies. And that was sort of put on the back burner for political reasons, and fetal stem cell and all of that stuff. But now that we have inducible pluripotent stem cell technologies, these research studies are becoming more invigorated and making their way towards clinical studies. So basically, in this case, we could take stem cells and plant them into the substantia nigra, and pretty successfully, they will regenerate dopaminergic neurons to replace those that have been destroyed. So really exciting possibilities in the realm of stem cell applications in treatment of Parkinson's disease. So keep your eyes on that as you move forward in your practice. So now let's switch gears a little bit and look at Huntington's disease. Huntington's disease is also a neurodegenerative disorder. This one has a genetic component that we have previously addressed, and it also affects the basal ganglia. But remember the dog I reminded you or let you know about as the thalamus, this example. So we have the dog in the thalamus, and he has some stimulation, dopamine. He's really happy about his little dopamine biscuits and is very excited. And then we had the leash running through the indirect pathway, sort of keeping him under control. Huntington's disease has more to do with a leash that gets a little bit loose. So now we have an excited puppy who's got his dopamine biscuits that is jumping all over the brain in somewhat of an uncontrolled manner. So Huntington's disease is also a movement disorder, but it is more on the opposite end of the spectrum than Parkinson's. So movements that are uncontrollable or inappropriate. So the leash is a little loose, signals are getting sent that perhaps shouldn't be sent. It tends to onset between 30 and 50 years of age, but can also be juvenile onset. You'll recall it is one of those expanding trinucleotide repeats. Here we have CAG with increasing generations, or the more generations that pass, we can expand this repeat, or this repeat does expand. And once we reach greater than, say, 40 repeats, it tends to get expressed as Huntington's disease, an autosomal dominant disease. So we end up with this mutant Huntington protein, which has additional glutamines, because CAG will end up coding for glutamine. And glutamines have pretty sticky tails on them. And so the more glutamines you get in the sequence, the more the tails stick together and the more the protein will clump. And that tends to result in the Huntington's disease. So we call this mutant protein mutant Huntington's protein, or Huntington. The symptoms of Huntington's disease are very progressive, and I've sort of outlined 3 stages here for you to consider. We've already covered that it begins with this sort of tremor and bradykinesia, might progress into some clumsiness, a little bit of restlessness, hard time sleeping, and occasional sort of twitches, as well as other muscle spasms right? So we'll see a progression of this to these spasms and muscle control issues actually affecting walking and talking, as well as performing tasks. Now, keep in mind here, this is a movement disorder. We're not seeing any dementia at this point. So individuals are perfectly capable of walking around, although it may appear a little bit clumsy. Many patients are still quite active and but exhibit this phenotype of lack of control of movement. It's only in the later stages that we start seeing dementia set in and communication becomes more difficult, even to the point where we start seeing uncontrollable outbursts. And of course, these uncontrolled body movements with dementia setting in also can be quite a rough situation. So Huntington's probably has the most severe of outcomes for patients as well as their families. Let's take a little closer look about what we know about the Huntingtin protein, or MHTT. It results in a multitude of different effects. First of all, it appears to be clearly most toxic in the brain. Anything that has a neurodegenerative effect clearly is going to impact the brain before anything else. But we also see that it's affecting not only connections between the basal ganglia, but also it starts affecting connections further out into the brain, which is where we see dementia setting in later stages in the progression. Again, this MHTT forms clusters in neurons. So we have a common theme with all of these disorders of aging. We have dysfunctional proteins being formed that tend to stick together and aggregate, forming clusters that get in the way of communication between the cells. We can see also the clusters get in the way of regular transcription. So other proteins tend to start to be degenerative proteins that are not in their normal form. And we also see there are mitochondrial disorders. So along with the neuronal disorders, we have disorders with transcription of other genes and then destruction of mitochondria, which result because somehow the caspase system is activated. And you probably, well, what does caspase get involved in? Caspase, you may recall, has to do with apoptosis. The mitochondria basically send out caspase to say, like, whoa, help me, rescue me, I'm toxic. And so then the immune system will be activated to come along and destroy them. So caspase is a signal that mitochondria use to signal that it's time for that cell to be degraded. So that would be a great explanation of why we see the neurons actually being destroyed in those connections. So let's finally consider what we do in treatment of Huntington's disease. And the sad thing here is there really isn't a lot in the option of treatment at this point. Relieving symptoms is really the most that we can do, and it generally will involve sort of a team of individuals because we have, as speech is lost, we might have speech and occupational therapists trying to help regain control of movements as much as possible, as well as psychiatry, because obviously if you're experiencing this, there's a lot of considerations for the health, your mental health or individuals' mental health. There have been some effective treatments with xenazine in reducing the chorea. The word chorea comes from a Greek word that means dancing. So individuals look like they have sort of this stiff dancing motion in the later stages. And so in the later stages, tetrabenazine is available for treatment of the chorea, but that doesn't really help with all of the symptoms. So in short, we have a multidisciplinary team approach to relieving symptoms as much as possible. But again, Huntington's is a pretty rough disease for everyone involved. So in closing this lecture, you should now have all the information that you need to know about Parkinson and Huntington's disease for studying for your exams. Thanks for joining me. Now let's take a look at summarizing some of the things that happen with mitochondrial dysfunction. What's going on in our mitochondria? We know that they are especially prone to DNA damage, and there's even a whole theory, the mitochondrial free radical theory, that suggests that the more we consume and eat, the more cell respiration that goes on, and thus the more oxygen is split in order to form H2O, right, which generates, when we split the oxygen, free radicals that can go around and do damage, and the more of that damage that's done, the more aged cells become. Now, there have been some challenges to this free radical theory, or expounding upon that free radical theory. It really depends on how you look at it. But this is how I would summarize that information. So it turns out that not only do we have the idea that, yes, indeed, mitochondria do accumulate free radical damage throughout their lifetime, and that becomes more with aging. But is it purely because of the increase in free radicals or oxidative reactive oxygen species? So the question is, do these species actually cause our deterioration, or what else might be going on? So more recent evidence suggests that perhaps the mitochondria, in response to stress, right, if they're not functioning that well, or if there's not enough energy around, we would upregulate the production of mitochondria such that there are many more mitochondria. And as a consequence of that, the more mitochondria are producing more reactive oxygen species, or 1/2 O2s, that are able to go around and do more damage. But we are also increasing the energy output, so that might counteract that. Anyway, so we are as a congruent thing, we are growing more mitochondria in each cell, and they are also producing more reactive oxygen species. And so perhaps the increase in reactive oxygen species, or ROS, is due to the fact that there are simply more mitochondria. Either way, the reactive oxygen species are causing damage and are certainly aligned with cellular aging. So just a little note on some of the newer research, but free radical theory is sort of the leading idea at the moment. Moving on to the next physiological trait of aging, you are probably very familiar with the idea of telomere attrition. Certainly, we have covered that telomeres shorten as cells go through divisions, and stem cells and cancer cells might have telomerase active, and so they can continue to grow or regenerate telomeres. However, most somatic cells lose that capacity, and you're probably familiar with the Hayflick limit. Hayflick limit suggests that there are a limited number of cell divisions that somatic cells can go through before the telomeres become short enough to start doing damage to the, subtelomeric regions of the chromosome. Once the telomeres have essentially run out. It's suggested that 50 divisions is about the maximum for any somatic cell. So after that, each cell division is shortening the ends of the chromosomes till we start actually nibbling away at the genes located on the ends of the chromosomes, which, as you can imagine, can have some manifestations in the condition of the cell. So telomeric attrition definitely associated with the aging cell. Epigenetic alterations. We've spent a lot of time thinking about epigenetics in the genetics courses. So epigenetics, again, above the genome. Traditionally, we consider epigenetic factors to be heritable changes that are not to the DNA, but to the way the DNA is packaged and how it is accessed. And we know that these epigenetic factors now can change throughout our lives and alter how and when various genes are expressed in the great variety of cells that we have. So these changes in chromatin state, in essence, are regulating the expression of genes. And we've definitely spoken about epigenetics being one of the factors that regulates gene expression. So when we consider epigenetic factors, I think that you could probably write down at least 3 different epigenetic alterations that could happen. So go ahead and try that out. And then let's move forward and look a quick review of what those are. First of all, we can have histone modifications. Histones are the cores around which DNA is wrapped, and depending on where those histones are located, DNA may be either accessible for transcription, or if it's wound tightly around the histone, inaccessible for gene transcription. These histone modifications can include addition of acetyl groups to the tails and changes in the wrapping of the histone tails, how tightly they hold the DNA onto themselves. And again, that lends itself to accessibility issues on the DNA. Another thing in epigenetic alterations we consider are how methylation patterns happen. You know that areas that are highly condensed heterochromatin, we have tight packing and again, lack of access to genes by the transcriptional mechanisms. There's a connection of DNA methylation patterns to the development and progression of Alzheimer's disease and how certain proteins fold in there. We will definitely cover Alzheimer's disease in a future lecture in a lot more detail. But either way, another epigenetic alteration. And all of these epigenetic alterations lend themselves to the concept of chromatin remodeling. So remodeling the areas that are condensed versus opened, and thus the regions that are available for transcription and translation. So in general, we see decreasing levels of heterochromatin associated with aging, and epigenetic remodeling or chromatin remodeling actually is changed by some environmental stimuli. And this is one of the very active fields of research at the moment, and how some of these environmental modifications to the, let's say, not DNA, the epigenetic factors can actually become heritable, and we're finding that they can. So that's a really exciting field, at least to me at the moment. Next, let's consider loss of proteostasis. Clearly, if we have changes in transcription rates and frequencies of a variety of different genes, then we have a loss of proteostasis, simply meaning that cells perhaps lose the ability to do quality control on their own protein production, and the protein production has gone awry because of these other modifications and insults to the DNA, reactive oxygen species, all of these different things. We see that the loss of proteostasis in beta amyloid plaques and tau proteins and such that we see in Alzheimer's and Parkinson's. We also see the disruption of protein homeostasis in cataracts. That's responsible for the cloudiness that occurs in cataracts. So again, disorders clearly associated with loss of proteostasis or homeostasis of the proteins. Things are getting a little bit dysregulated. So accumulation of these damaged proteins, as we see in Parkinson's and Alzheimer's disease, clearly cause some phenotypic effects, even can be toxic. if not just interrupting signaling. Next, we look at deregulated nutrient sensing. This is definitely associated with aging. Even if we think of the individual as a whole, they are taking in less nutrients than they would be in more or in younger cells. So deregulated nutrient sensing involves multiple different pathways. Right now, we're considering that on the cellular level. So each of the cells in our body, particularly when we think about metabolism, cellular respiration, and the creation and storage of fuels, right? If we have fat to be in storage or fat to be released from storage, we will see changes in free fatty acid levels, as well as visceral adipose tissue and total body fat compositions. All of these change as we age. We also see some of these nutrient sensing pathways changed in muscle tissue, changing the rate of fat oxidation versus protein synthesis. So again, an issue with storage and release of nutrients. Things are just not timed as efficiently as they used to be. The liver, of course, also involved in these nutrient sensing pathways. So this is sort of a paradox because when we consider that older adults tend to intake less food, they are also extracting less from that food. Now, there is this hypothesis that I think I alluded to it earlier or mentioned it earlier, that the more we eat, the more reactive oxygen species that we produce. There's a lot of research showing that older adults that are consuming less, or anyone consuming less, actually, you don't have to be older, consuming less creates less reactive oxygen species and thus less cellular damage. It's also well known that as we increase our exercise, we produce our own endogenous molecules that act to counteract that. So we can get rid of those reactive oxygen species. So as there is a decline in nutrient sensing, this is kind of the paradox. As there is a decline in nutrient sensing, we also tend to age the cells more because they're getting less of the necessary nutrients in the right balance. And so that creates us a situation where one thing sort of feeds the other. And obviously, very active areas of research presently in cell biology. So next, let's look at altered intercellular communication. As you could imagine, well, think about for a moment all of the things that you know about cell communication. What cells come to mind when you think about cell communication? And what types of signaling come to mind when you think about cell communication? I'm sure that a plethora of things came to mind as you think about all these different protein cascades and signaling between cells, signal molecules activating other cascades within cells. So lots of different things become less efficient as cells age. Particularly, you might have thought about neurons because they're definitely signaling cells and neurohormonal signaling that occurs between neurons. So definitely as cells age, this signaling becomes deregulated, and we see certain evidence of this as we aggregate cells in whole organs in the aging process. In addition to the signaling between neurons, one of the areas that we see a lot of action, so to speak, is in inflammatory reactions. The immune system cells are not necessarily talking to each other in the way that they would be in a younger cell lineage. So we are deregulating intercellular signaling in that manner also. So when we consider the inflammatory reactions increasing, we also can see that they are going to deregulate cell signaling, including things like adrenergic signaling. So these things become slower. Renin-angiotensin system, you should be familiar with most of these from your understanding of physiology, as well as insulin and IGF signaling, and immunosurveillance. All of these categories of signaling become deregulated based on some of the inflammatory reactions. Now, another concept that we haven't really spent any time looking at is the communication of cells through gap junctions and the concept of bystander effect, which is sort of like contagious aging, where through gap junctions, as you know, gap junctions allow passage of actual free passage of ingredients or materials between cells. And so what is in the environment of one cell will move through a gap junction into another cell. So you could see how essentially we're letting these qualities of aging pass from one cell to another. And it's noted that when one cell ages, cells in its neighborhood tend to age also. So the bystander effect is something you should be familiar with in aging, contagious aging. That's how I will keep that in mind.